I am Kody Rominger. I am the program manager of botany research at Utah Valley University. And my partner in this project and many of the other projects we work on is Susan Meyer, who you'll hear from right after me and after break. And what I'm going to talk about are specifically about a project, a research and active management strategy to reduce extinction risk for endangered bear poppy. That is a mouthful. And that's a project that's has all these different collaborators here. Everybody has a little piece of it. Seems like it's a huge project, but specifically, let's move on here. So this is all the different various objectives that we have in this project, which is why we have so many different partners. And I'm just going to talk specifically about a couple of these, which one is the range-wide drone census, and the other, depending on how much time we have, some long-term monitoring, as well as the reproductive output. It's on the ground part of the monitoring that goes hand in hand with the drones. So I'm just going to talk about those top three and maybe one more if I have the time. But I have this little graphic here. I just wanted to show kind of, with all of these different objectives we have in the study, the first 5 or 6 are dependent upon the foundation of the drone census. And the drone census ultimately ends in one of the products is a species distribution model that we're also going to use to kind of the big completion of the project, which is an augmentation of existing populations and introduction studies in areas that aren't currently occupied for the poppy. So drone census foundation, ultimately ending in, hopefully we can get areas of introduction, get new populations established. So for the range-wide drone census, we did exactly what it says. We flew the whole range of the known dwarf bear poppy populations, as you can see here in the gray or in the yellow polygons, which automatically default to red bluffs because it's kind of traumatic still from the experience of flying and spending so much time at red bluffs. That was really a huge area. Took a long time to do, but we did do it. We red bluffs, white dome, this little Val Springs population, white dome, Webb Hill, Beehive Dome, Warner Ridge, and this is Shinabkaib. We did not fly this population here because it's on private land, so we haven't been able to get access there yet. And we do have one little tiny speck of Warner Springs that we got to get this year. So all in all, we flew over 9,000 acres. We collected 1.5 terabytes of just raw drone imagery. We then developed an imagery processing workflow where we took all of this. We're still in the process of doing that, but so far we've taken a lot of this imagery, processed it into orthomosaics and DEMs, and we've already added another 2 terabytes in additional data just for the outputs of those processes. We then used a manual/visual detection method to go through some of these orthomosaics, like at White Dome here, where we went through and manually marked each of these poppy points. And that took a long time, as you can imagine. This is 800 acres. So just based on how much additional imagery we had, we knew we needed to come up with a way to be more efficient in detecting poppies. And so we developed an additional artificial intelligence model, or a deep learning model, to do the poppy detection for us. And ultimately, we're going to have as our final product a map across all of the range that has individuals greater than 6 centimeters in size mapped all across each population for the entire known extent of the bear poppy. So that's going to be pretty powerful. The manual detection, I we got published in 2019 with remote sensing. So specifically with the AI poppy detection part, we used the model Yellow V5, which stands for you only look once. This is just it's a object detection model, one of several that we decided to go with. And we trained the model first by giving it images similar to this one on the right here, where we had to go through manually mark poppies and draw boxes around poppies in hundreds of images. And we fed that into the model, and the model, it went through thousands and thousands of iterations, and we had to fine tune and give it more imagery. And we gave it images of bounding boxes of poppies. We also gave marked other things. So there's not really a good example in these photos, but we marked things that looked similar to poppies, but were not, so that we could give the models a way to distinguish similar looking vegetation, which greatly improved the accuracy of the model. So after training, we ran validation on imagery that the model had never seen before. And that the outputs is what I have examples of here, where the model on the left here, the model went through, this is its detections, called all of these poppies, and then gives a confidence level. And we go back and verified that each of these were or were not poppies. In this case, on both sets of examples here, each of the detections that the AI made were in fact poppies. So then we went a step further, and we took the same model, we took a different set of imagery, and we, over 12 different areas, we tested the accuracy of the model to a human worker doing it that had also never seen the imagery, and found that the model was able to detect the poppies 83% accuracy versus the 67% accuracy from the human worker. So it's better. And also we were able to significantly decrease detection time versus the manual detection time. So for example, we developed this methodology using the Webb Hill imagery, which is 630 acres. When we ran the AI model on it, it took 147 minutes, just under about 2.5 hours. Whereas when I went through White Dome, and it's a little bit bigger, 800 acres, it took me to mark all of those poppies individually. It took me in the neighborhood of 50 hours. That's my best guess. It was probably a lot more than that, but we're going to go with 50 hours. But clearly there's a gigantic difference in how fast an AI can go through imagery compared to a human worker. So that was a big achievement for our team, and we were able to get those results and that method published last fall in the Drones Journal. So then again, our last step is we want to get all of these populations mapped to individuals across the whole range of the species, and we're still working on our protocols for getting that rolled out. So I still have a few minutes. I want to talk about kind of when you have census data for a population, you can use it to determine kind of where you want to put long-term monitoring plots, or at least that's how we did it. So we had this census data that I did, and others helped with manually at Beehive Dome, White Dome, Schnaub Kaib, a piece of the Red Bluffs population, and at Val Springs. And since we had that census data, we went and we chose 9 different plots within the populations based on the densities that we were able to generate from the individual census data, individual plant data from the census. And overall, we're looking at more than 4,000 individuals across all these populations so far. And for each of those plants, we measure diameter, which we have an example here. So we do this manually. We just look for the maximum, what we think is the maximum diameter, and we just use an arc, and we use the measure tool, just draw a line, 42 centimeters, enter it in the spreadsheet, and go on. And so after the first year, then we could start looking at survival. And then we finally look at whether the plant is flowering or not. So we have 9 monitoring plots at each of the populations, and each of those monitoring plots are 0.6 hectares for a total of 5.4 hectares per population that we're looking at so far. We do have plans to expand that in 2022 as we get additional census data in. And we want to do this for the remaining populations as well, but in 2022 for Warner Ridge, Webb Hill, Warner Springs, and some additional areas at Red Bluffs. We have Tonopah already, but we want to add, since it's so large, we want to add a couple of different areas of monitoring out there. This table down here, I wanted to just show without getting into any of the analysis or interpretation, but this is what the kind of data that you can get by monitoring over multiple years and 5.5-ish hectares. Each of these is a plot, and this is representative of this beehive dome population. But this is what we started with in 2019, the initial cohort, which was bulked up due to a recruitment event in 2017, which is part of the Poppy's life history strategy. So we had a lot of plants to start with. And then in 2020, for each of the plots, there was a decline. And then in 2021, there was a huge decline. So survival from 19 to 20 was 54%. From 2020 to 2021, it was only 4%. For flowering, it was similar across both years in the 90s, in 2019 and 2020, and then 2021, it was only 60%. But yes, the diameter, I guess I was going to say, just take that with a grain of salt, because there's only 23 plants total in 2021. So that's not necessarily a good representation of, of flowering, but that is what it was for that year. So 2019, you have, this is the average diameter. You have a little increase in 2020 and a little decrease in 2021. But the growth from 2019 to 2020, you had some growth here, but from 2020 to 2021, you have a huge drop off. And it's kind of weird to say negative growth, but these poppies grow in rosettes, so it's just a matter of, many of the rosettes have died off in some of these plants, so it's negative growth. But I just wanted to show this as an example of kind of the power of what you can start learning, looking at different populations over multiple years and following so many different individuals. And I'm probably not going to have a lot of time to get into this, but on top of that, each of these populations, we also have it on the ground addition that we do to where we look at the reproductive output. And in the field, we also take the diameter, number of flowers, the fruit set, and then back in the lab, we look at the seeds per fruit, fill, and the number of aborted and unfertilized seeds. And we did a little comparison where we looked at the drone imagery versus measurements of diameter versus what we got when we looked at them on the ground. And that's just displayed here, just a little aside. But we're very good at, very consistent, I should say, about how we measure in the field, and looking at the same plant, what we would measure from the drone as well. It's very similar across. I think we had 100 plants that we tested this on. I'm not going to go into all of this. I'm starting to run out of time, but kind of the take home here is, we add a little not a little, but we add a on the ground component to reproductive output with the drone based monitoring. And you can get this kind of data then where we can look at the seeds per fruit over each year and see what the differences is there. And it's just another level of data for this long-term monitoring / demographic monitoring work. So just an example of that. And this picture was just a little example of how small the seedlings are and why you really can't do anything on the seedling level, because they're just, you're lucky to even see them. Yeah, let's move on. And then lastly, just really quick, just an example of what else we're doing on the monitoring plots with this drone imagery. So we're doing some habitat and disturbance evaluations where this is the monitoring plot boundary on the right here, and the monitoring plot itself on the right hand side where the marked plants are. That's 2019. Looked at the same area in 2020. And this is with a better drone, with a little bit better resolution imagery. And we still have the same plants marked. They're all there still. There's one more that we could add. And then 2021, same area, all the plants are gone. And you can start to see maybe why they were gone. You're looking at this rodent disturbance. So we're figuring out ways to analyze this and try to figure out what it all means. And if it, I'm trying to think of the word, if it goes along with the increased Rominger invasion that we've seen out in poppy habitat as well. I don't have time to get into all that. So another thing though is that you can look at it for disturbance. You can look at these areas and see that where should be intact crypto crusts. You can see that all these bare patches are where cows have walked through probably over many years. This isn't one season of disturbance, but over many years, you can see it makes a big impact on the habitat, and where this is especially important when we're talking about the endangered and rare plants of Utah. So I think that's probably all the time I have, but I'd like to thank or acknowledge the Nature Conservancy in Elaine, York, Fish and Wildlife Service, Jenna Lewinson, the BLM, Stephanie Taylor and John Kellum out of the St. George Field Office, and Aaron Rowe from the State Office, ESMF, USU, Mindy Wheeler, the Washington County HCP, Cameron Rognan and his team, as well as Allison Denettis for her work on the RO studies, and the drone pilots, Sid Houghton, Mason Freeman, and Eli Harton, who helped with capturing a lot of the census data. And then, of course, I have my famous picture of Dr. Meyer and Dr. Van Buren. I love this, my two mentors. Both these people mean so much to me, but great mentors, great people. But one last thing, when I think of Renee, though, I think of astragalus homegrown Orem. That's how she got me started. I don't think of poppies. So I just wanted to throw a picture of astragalus in there. And then it's just a couple of, oh, that actually is Renee's picture too. And yeah, that's it.